All right, I think we're ready to get started. I'm Chris Lorenz, Professor of Biology at Thomas More University. I want to thank you for coming tonight. This is the 4th year of our Marine Biology and Conservation Lecture Series. How many people have made it to just about every lecture so far? Excellent. And how many people, is this your very first lecture? Awesome, excellent. So we're all in for a treat. I'd like to thank Eric Rose. Eric is the Executive Director of the Newport Aquarium and Vice President for Hershey Entertainment. We want to thank him for his hospitality and opening up the Riverside Room tonight. So please acknowledge Eric for that. We'd also like to acknowledge the Marine Biology Lecture Series Committee chaired by Dr. Stephanie Snyder, who happened to be our last speaker, Aaron Schultz, Judith Chris, Lom Film was in the back, and I may be missing a few others, but if I am, I apologize. We'd like to acknowledge and thank the committee for their hard work in putting this together. And now it's my pleasure to introduce Dr. Kathleen Jagger, the president of Thomas More University, who's a biologist by training, microbiologist and epidemiology are her background, but really an environmentalist and a lover of nature. And so we're pleased to have her welcome us on behalf of the university. Dr. Kathleen Jagger.
Wow, thanks, Chris. I appreciate that. On behalf of Thomas More University, I do want to welcome everyone to tonight's presentation in the Marine Biology Lecture Series. Again, thanks to the Wave Foundation, thanks to Newport Aquarium for this partnership, thanks to Stephanie Snyder for coordinating the Marine Biology Conservation Lecture Series. This is really, truly a pleasure for you. If you've never been to one before, you know, strap in, it's going to be great. So, as you may know, Thomas More University is the Catholic liberal arts University of the Diocese of Covington, Kentucky. Inspired by the Catholic intellectual tradition, we challenge students of all faiths to examine the ultimate meaning of life, their place in the world, that's important tonight, what's your place in the world, and responsibility to others, not just other humans. So we do this by developing and sustaining challenging academic programs, undergraduate, graduate, professional programs of study. And those are always marked by superior teaching and scholarship within the Catholic intellectual tradition. And I say that because I think tonight's going to be, no pressure, Dr. Whitney, but tonight's going to be another example of superior teaching, right? Exemplified by this lecture series. Teaching about a species that has its own place in the world, which he's going to talk a little bit about, I think. So tonight you're going to hear about sharks, right? What's the first thing that comes to mind when you think of sharks? Baby shark? Do we have any students that want to come up and do a rendition of Baby Shark? You want to come up and do Baby Sharks? Yeah, let's hear it. Give it. Sure.
I need the younger kids to help me.
Join in. Let's
come on. Come on. Ready? You guys got this? Oh, okay. Ready? Here we go. Baby shark, doo doo. Come on, get into it. Come on. Baby
everybody else join in if you know
baby
it.
Shark. Daddy shark, doo doo doo. Daddy shark, doo doo doo. Daddy shark, doo doo doo. Daddy shark. Grandma shark next? I think so. Okay, let's do Grandma shark. Grandma shark? Sure. Okay. Grandma shark, doo 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 doo. Grandma shark, doo doo doo. Grandma shark, doo 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 doo. Grandma shark. Grandpa shark, doo 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 doo. Grandpa shark, doo 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 doo. Grandpa shark, doo 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 doo. Grandpa shark. Is it swimming? Sure. Shark is, or I don't think we're gonna do shark. Let's just, what are we doing? That's good. Okay.
So some of the other things that you might be familiar with is Shark Tales, Sharknado, right? My grandson's, one of his favorite movies is Megalodon, or just, you know, think about a day at the beach gone wrong. So clearly what we have all seen in the news and our entertainment industry about sharks is not good news, right? But with over 400 species of sharks, you know, it's really not fair that we generalize the stereotypes from these movies and tabloids to all sharks. And so maybe you'll take home a different view of all that tonight. And I don't think that those generalizations even apply to what seems to be the most fascinating species of great whites. In my mind, sharks are the snakes of the ocean, and I really love snakes. But snakes get a bad rep too, right? So almost always getting bad press, but being really truly essential to the ecosystems in which they live. So I'm excited about hearing more about sharks tonight. But before I do, I want to tell you about one other event. So mark your calendars, make a mental note. On March 16th, on Thomas More's campus, our own Dr. Stephanie Snyder is going to be giving one of our observatory talks. So why is a marine biologist giving an observatory talk? She's going to be talking about fish migration from space. How do you study it, from satellite system? You're all invited. Our observatory talks are on Saturday night, so March 16th is a Saturday. Starts at 8 o'clock in Steigerwald Hall. And then, weather permitting, if the sky's clear, you get to go to the observatory and look at what's really cool in the night sky. So those of you that are science geeks, generically, not just about marine biology, we welcome you to come and join us for that observatory event. Again, March 16th at 8 o'clock. And so, with great delight, I'll turn it back over to Chris so we can listen to tonight's expert, Dr. Whitney.
Nick, you have a tough act to follow with that baby shark there, so the pressure's on. One more housekeeping item, and it's an exciting one. Each lecture series we do a raffle. For those that are astute enough to register early, your name is entered into a raffle, and then Dr. Snyder, our marine biologist at the university and chair of this committee, drew a name randomly, and she is going to announce the winner of the raffle, who gets to win what?
They get to win a behind-the-scenes shark experience for 4.
Nice. And our lucky winner is?
And I want to say, we did not draw this. We did this in true science fashion, random number generator. Lowest number wins, and it was Madison McVeigh.
Excellent. Congratulations, Madison. Remember, there's 4. 1. All right, so we are indeed in for a pleasure tonight. I had the pleasure of meeting Dr. Whitney a few months ago when Eric Rose brought him out to the field station, and his passion, his enthusiasm, his wisdom, and expertise were certainly evident from our short conversation together. And Nick is a product of a small liberal arts college himself. He attended Albion College in Michigan, a really elite private school much like Thomas More here in North Kentucky, and he got involved with shark research at an early age. As an undergraduate researcher, he connected with a professor, one who was involved with sharks, and the rest is history, as they say. So after graduating from college, he attended the University of Hawaii, tough place to be, for his master's. He loved it so much, he and his wife were there for 9 years, 8 or 9 years, is that right? Got a master's and Ph.D. at the University of Hawaii. And then when you are a prolific researcher like he is, you go out and search for postdoctoral opportunities. And with his accomplishments, he got a very prestigious one at the Moat Marine Laboratory in Sarasota, Florida, one of the elite marine biology labs in the country. So he moved from Hawaii to Florida, and he started his postdoc, which transitioned then into a job. And I think that's where they spent another 8 or 9 years down in Florida as they grew their family, 1 daughter and 2 sons. And then we were fortunate to have the pool of family to come back up here, and they live nearby in Blue Ash, where their in-laws and his parents are in Michigan, and then they have some relatives here in town as well. So the good fortune of that is ours, in that Dr. Whitney approached Eric to have a joint appointment with the Newport Aquarium and the New England Aquarium. So Dr. Whitney's a busy guy, spends his time here, spends his time on the East Coast, and then of course spends his time out in the field. His next research expedition will take him back to Florida, where he'll continue his work on sharks. And so it's my pleasure to welcome Dr. Nick Whitney as our speaker for tonight.
Thank you. Thank you very much, Chris, for that introduction. Thank you all for coming. Thank you, President Jagger, for stealing my thunder on the Daddy Shark song. That's how I was going to start my presentation. But, I don't have to do that now. The kids did it much better than I would have done. So I also just wanted to start out by, sorry, there's my title slide. I also wanted to start out by just acknowledging the fantastic relationship we have here between Newport Aquarium, the Wade Foundation, and Thomas More University. I've been involved with all three of these institutions over the past couple of years, and culminating with this week where we had a great activity with the Thomas More University students out at the field station on, I think, Tuesday night. And also we had some folks there from the Wade Foundation and Kelly here from the aquarium, our new curator, answering questions and talking to the students about what it's like to have a career in marine science and marine biology. And it's been very rewarding, and I'm thrilled to be here. So now, as you can see, I'm going to talk to you about understanding the secret lives of sharks, which it turns out is a hard thing to do. That's because we can only, if you think about it, we can only really observe sharks directly in the most perfect, pristine conditions. We need clear water on a bright sunny day, and we need the sharks up near the surface, which they're almost always up there because we've lured them up there with bait or chum or fish blood. So anytime you've seen sharks, whether it's on Shark Week or on TV or whatever, you've almost never seen them doing natural behavior. You're going to see a little natural behavior tonight, but for the most part, I'm going to talk to you about the different tools and techniques that we use to learn about the secret lives of sharks, because for the vast majority of time, we can't observe them like this. To start out, though, I want to give you a little bit of sort of shark history, going all the way back to the beginning, the beginning of Earth, 4.5 billion years ago. For most of the history of Earth, there was life, but nothing you would recognize as life. It was just microscopic until about 540 million years ago, when something happened called the Cambrian Explosion. And I'm just going to zip through this. You don't need to memorize these numbers. There won't be a quiz at the end. But starting about 540 million years ago, we started to see the development of animals that you would recognize as animals, and it started mostly in the ocean. And about 450 million years ago is when the first sharks appeared. If you could go back in a time machine and go snorkeling in the seas of the Ordovician, just before you were eaten, you would see something that looked like a shark. But this is several million years before we even had trees on land or insects, way before Jurassic Park happened, or the Jurassic period, way before the dinosaurs. And of course, millions of years before any of the animals on Earth that we would recognize today as our common animals. And of course, we humans came along only in the last second of evolution. The first humanoid or hominids came along about 2.5 million years ago. This is not a human, or this is not a Homo sapien. This is another species of human, Homo erectus, that actually lived for about 2 million years. But let's talk about human history for a second. We tend to think of it with this diagram, right? We know that we have common ancestors with the great apes. This is actually a misrepresentation. It kind of implies that we started out here and then we sort of got taller as we evolved, right? In fact, we now know that there were actually several species of humans living, maybe as many as 6 species of humans living throughout the Earth at one time. Homo erectus being one of them, the Neanderthals, of course, many of you have heard of those. We just found out recently that from those of us who have European or Middle Eastern DNA, we probably also have some Neanderthal DNA in you. So, but one thing that all of these species of humans had in common is that we were all, for the vast majority of time that we've been on this earth, we have been part of the food chain. We were getting eaten by all kinds of animals, just like every other animal. So think of anything: lions, tigers, bears. Oh my, yeah. Anything you can think of, plus a huge list of species that are now extinct that you never even thought of before were also eating us. All kinds of cave bears and and cave tigers and eagles would eat us, and various snakes. So this is really the conditions under which our species evolved and what we're really used to. It's only been in the last couple thousand years of our history that we've eradicated all of these predators and can now walk around mostly free of worried about being eaten. But if you think about when was the last time you actually have felt any risk at all that you might be captured and eaten by a predator, it was probably when you were swimming in the ocean. And this is something that Peter Benchley and Steven Spielberg realized back in the 70s with the book and the movie Jaws. And I'm just going to show you a little bit of the opening scene. I'm going to mute it about halfway through, just so we don't scare anybody too much, because there tends to be quite a bit of screaming. But for those of you who haven't seen this movie, this is the opening scene where we just have a woman out swimming at night in the ocean, which no one ever does now that after this movie. Trying to let you hear the music of this. This is the part where I mute it. So this is actually an incredibly unrealistic sequence. Getting bitten by a white shark is not like riding a jet ski. It's not how it works. But you can see this is obviously a terrifying scene to watch, especially for young children. I know I personally was shocked when I found out that this movie is actually rated PG, which is why I'm comfortable showing it to you, as long as it's muted. So yeah, that was in 1975, and I don't have a picture of myself after I first saw this movie when I was a kid, but growing up in Michigan, this is what I knew of Jaws. What I do have is a picture of my kids when I showed them this movie. This is right after the opening sequence. You can see they're terrified. Even Margot was trying to escape the room, and she couldn't even walk yet. So that gives you an idea. In fact, if you look at the expression on Nigel's face here, you might recognize the same expression that you saw in Homo erectus. This is a primitive instinct. He's remembering what it was like for us to live in fear of predators for all of those years. This is a picture of me. So after I had seen that movie growing up, whenever we went on vacations to the ocean, I wanted nothing to do with swimming. I stayed up on the beach, playing in the sand. As I got older, I played a lot of beach paddleball, and went on a lot of boardwalks and walked for birds, and never wanted to swim. The only thing that prevented people from teasing me for being afraid of the ocean was my fashion sense. How could you make fun of that? But I started to try and learn about sharks. I started reading books and scientific volumes about sharks, because I wanted to figure out if there was a way I could go in the ocean without being eaten. And the more I read about sharks, the more interested I became. I turned into a shark nut. But some people had a different reaction to Jaws. A lot of people saw that movie and read that book and decided that, hey, we need to do something about these sharks. The idea that the only good shark was a dead shark became much more popular. Fishing for sharks became more popular. And that's part of the attitude that was going on here. This is Hawaii in the early 1990s. They had a couple of years where there had been a larger than average number of shark attacks. And the state started this policy of going out and catching and killing as many sharks as they could whenever someone got bitten. They would, if there was a bite off of Waikiki Beach, they would go out there the next day and kill as many sharks as they could off Waikiki Beach. This was when science came in and shed a little bit of common sense on this. There was a group out there that started tracking tiger sharks using these acoustic transmitter tags that send out a signal underwater and allow you to follow the shark from a boat. So this is the island of Oahu, and these different colors just represent different depth of water. But if you look at these lines, these are tracks of individual sharks that they tagged and followed from a boat. And you can see they almost all go straight offshore. So this research revealed that within the first day or two after a shark is caught off of the beach, it's more likely to be 100 miles offshore or even at another island than it is to be at the beach where that bite took place. So this research put an end to that shark culling that was happening out there. At the same time this was going on, or close to the same time, I was actually an undergraduate at Albion College in Michigan, as Dr. Lorenz said, and I was using very similar technology. I was studying, with my professors, I was studying nurse sharks and using these acoustic tags to track them. You remember that picture of the tiger shark? It's literally the exact same tag that we're using on both species. In my case, I was tracking these sharks from a kayak. My job was to paddle a kayak around all day and stick this underwater hydrophone, this underwater microphone into the water and listen for that ping from the shark's tag. And when I turned the hydrophone in the right direction, I could tell by the sound of the ping which direction I needed to paddle to follow the shark. So I did this all day long, every day. I thought it was the greatest job in the world. And only now do I realize there's a reason why the undergraduate interns do that job and not the professors. But fortunately, when you paddle a kayak around all day, you get pretty nice arms. It was definitely selling tickets to the gun show on that one. But the nice thing was, having this experience tracking sharks in the Keys made me very well qualified to get into graduate school with that group in Hawaii that was tracking sharks. So I graduated from Albion College and went to the University of Hawaii in 2000. And once I got out there, immediately started working with that group to help tag these big tiger sharks. Hawaii is a great place to go to find sharks, and especially big tiger sharks. We would catch them and bring them up alongside our boat. A lot of times we'd roll them over onto their backs when they go into a sleep-like state called tonic immobility that allows you to do just about anything you want in terms of measurements, attaching tags. We can even perform minor surgery when they're in this state. And then we would attach our tags and roll them back over, take the hooks out, and send them on their way. Here's a picture of the largest tiger shark ever measured by scientists. This one was 15 feet and 1 inch long. And you can see we caught it out of a boat that was 17 feet. So here you can see it's literally as big as the boat, which is what fishermen always say. But in this case, it really is almost as big as the boat. And I took this picture myself by diving down and looking up, and almost got confused and disoriented as I floated back to the surface and almost swam right into this tiger shark's mouth, which would have been about the dumbest way that any shark scientist has ever met his fate. But it would have been a dangerous thing because these tiger sharks have incredibly sharp teeth and powerful jaws. This is what they can do just to each other. So occasionally we would catch a shark on the line and pull it in, and it would only be half of the shark. This tiger shark was probably about 8 feet long before it was caught, and then before we could pull it in, a larger tiger shark came along and just bit the whole bottom half of it off. If I were to give you a shark carcass and a really sharp fillet knife, it would take you a lot of work and time to cut through a shark this size. They have very tough skin and a lot of muscle, and this tiger shark was able to do it almost in one bite. But fortunately, we could get in the water with them when we released them and swim with them and take pictures. They never bothered us at all. They just wanted to get on with their lives. So we got cool pictures like this. But the species I worked on for my PhD dissertation was the white-tipped reef shark. This is a species that's very commonly seen in Hawaii because it can rest on the bottom and breathe. Some of you may know that many sharks have to swim constantly in order to pump water into their mouths and out over their gills. White-tipped reef sharks and several other species can actually rest on the bottom. And so it's easy for divers to find them in caves or under ledges like this. So again, I tracked them with that same technology, those acoustic pingers that tell us where the sharks go. This time we surgically implanted them in the shark. And they let me have a motorboat to do my tracking. I didn't have to paddle a kayak around. I was a little bit smarter now because I was in graduate school. So I asked for a motorboat. Another thing we did to track their movements was we used photo identification. You may have noticed that the sharks have spots on their side, and each shark has a unique spot pattern. And this was during the time when digital cameras were just sort of becoming commonly available. So there were a lot of divers now suddenly swimming around with cameras, and we took advantage of that and built our own database where we would match up their spot patterns and figure out which sharks were living in what caves and for how long and where they were moving to. Another thing I looked at with the species is population genetics. So even though the species lies around a lot and doesn't seem to move very much, they're found throughout the entire tropical Indian Ocean and Indo-Pacific, to the Hawaiian Islands, and even over to the Eastern Pacific. They've crossed to the Eastern Pacific barrier, which is pretty amazing for a shark that spends most of its time lying around. It doesn't have a planktonic larval stage like a lot of fish do, where their larvae can be carried out on the ocean. They give birth to live young, and they usually only have on average about 3 offspring every 2 years. So they don't reproduce very fast. And so, of course, I always say this is the same reproductive rate that my sister has, and she rarely gets out of the house. Which I say that because my sister is here, of course. And that joke was a lot funnier before all these kids got older and now live in the same town as me and track mud through my house and food. You guys. So, it was a big mystery as to how these sharks got so far dispersed and how much genetic mixing was happening between these divergent populations. It turns out there's very little mixing between these populations, and most of this dispersal happened fairly recently. But in order to find that out, I had to get DNA from these sharks. I had to find a way to get tissue samples. And rather than trying to catch every single shark on a hook, because white-tip reef sharks don't take a bait very easily, I developed this technique, which is these biopsy tips that go on the end of a spear gun. So I would swim up and shoot the sharks with a spear gun, and they would remove just a little plug of tissue, and to do that looked like this. This is me free diving down with my biopsy gun. In a second here, you'll see the shark. Like I said, they like to hang out in these caves and coral ledges. If you watch closely, you'll see, there you go. You could tell where I got my sample, couldn't you? So, and this is what they did almost every single time. When we took our sample, they would swim away. They didn't know what that was. It turns out sharks don't like getting shot with spear guns, and I probably wouldn't either. But then they would circle around, and eventually it would come back in the exact same cave. They didn't seem to get very disturbed, and we would be able to monitor them with our photo identification and tell that the little bit of a wound that would make would heal up very quickly. Sharks have incredible healing capabilities. The other thing we did to get a sample was we used something that I learned from the Midwest, these pig ear notchers. If any of you have spent any time on a pig farm, they use these things that are sort of like a paper punch, except instead of punching a hole, it punches a triangle out, and we can use those instead of marking pigs' ears. We would sneak up on these sharks that were sleeping on the bottom and clip a piece of their tail, so we could get a tissue sample that way. If this sounds like you're kind of asking for trouble, you're right. I did get bitten doing this. This is the shark that bit me. This is the exact one. I took a picture of it beforehand, and there's a long story here. It was my fault, but it was more my dive buddy's fault. I won't go into the story, but I do still have all of my fingers, and the shark was okay too. I did get the tissue sample, by the way. I did get it, but he sampled me as well. So once I was done with that work with the white-tip reef shark, it was time. I was finally finished with graduate school in Hawaii after 8 years, 8.5, almost 9. I finished both my master's and Ph.D. And for you kids in the audience, I always talk about how I did 4 years in college and then 8 years in graduate school. So if you do the math on that, that's like being in 24th grade. Finished. So can you imagine? Yeah, I know. So at that point, I was fortunate enough to be offered a postdoctoral scientist position at Mote Marine Laboratory in Florida. By that time, my wife and I had 1, and shortly after 2 children. Both of them are here tonight. There's more on that coming. And that's also the same time I started to use something called an accelerometer. So whether you realize it or not, you probably own at least 1, if not more, accelerometers. You probably have one on you right now, because they're found in almost all modern electronics, from digital cameras to smartphones. When you're looking at a picture on your phone and then you rotate the phone and the picture rotates, so you're still looking at it upright, it's because the accelerometer in the phone is detecting acceleration from the Earth's gravity, so it knows which way is up and down. Likewise, for any of you kids who have played the Nintendo Wii, or any of you adults who have played the Nintendo Wii, it's an accelerometer in the Wiimote that's tracking the movements of your arm. And for those of you counting your steps with a Fitbit, it's the exact same sensor, that accelerometer, that's tracking the motion, the acceleration of your arm, and counting how many steps you take. So this technology was put into wildlife tags to attach, and I got my hands on one of the first wildlife tags ever made that had an accelerometer, and they sent it to me and told me to test it on some of the animals that I work with. And of course, the closest animals that were nearby were my kids. So I tagged them, I put accelerometers on their shoes, but then later I had more kids, and so I had to make another video, just to be fair, that included all of them. So if you look at their shoes here, you'll see all three of them have accelerometer tags attached to their shoes, tied to their shoelace. And I'm going to show you a video in a second here that shows you what kind of data you get from different movements with these accelerometers. But before I started, I just want you to look at these faces and tell me which one you think did the best job of following my instructions for this experiment. It turns out it's the one in the middle. He did the best job. That's because Margot was just a little bit young during this experiment, and then Owen was just too cool to really have much interest. But the data below, so I want you to watch Nigel's foot here that has the accelerometer, and the data below actually correspond to the movement of his foot. So in the beginning here, he's mostly holding still, so you get a flat line in the data. Now he's taking small steps, and you get small spikes. And then in a second, he's going to freeze. I was yelling, freeze. There he goes. So he has an itch, but this foot's holding still, so you get a flat line. And then you see there's bigger spikes here, so you know he's about to take some bigger steps, which there we go. He takes bigger ones. Now he freezes again and you get a flat line. And now you see something really exciting is about to happen. Look at these big spikes. So that's what I'm doing. Just go crazy. When you tell Nigel to go crazy, he goes crazy. So you can see a lot of people, when they hear acceleration, they think I'm talking about speed or velocity. In fact, acceleration is a change in speed, a change in velocity, which is why you get these spikes with each step. So the next thing I did was actually put them on a shark. And we did these white-tip reef sharks, which I told you can rest on the bottom. So see, you get spikes when the sharks are swimming and flat lines when the sharks are resting. This sounds like pretty basic information, right? It doesn't sound like some huge epiphany, but when you think about it, when I was back tracking sharks using those acoustic pinger tags, and the tag isn't moving, I'm just getting a ping in one place. I don't know if the shark's resting in a cave, just sleeping, or if it's involved in some kind of feeding frenzy and just staying on the same reef the whole time. Either way, both of these behaviors would give me the exact same ping from my tracker tag, but now that I had these accelerometers, we could actually tell what the sharks were doing instead of just where they were going. So that was a huge revelation. But I wanted to take these tags back to the place where I started doing research, the Florida Keys, a place called the Dry Tortugas, 70 miles west of Key West, which is a unique place because it's one of the only places in the world where you can observe shark natural behavior in clear, shallow water on a predictable basis, and that behavior is mating behavior. The sharks come in here every summer into shallow water to mate. The females aggregate. You can actually wade right in, and if you're sneaky, you can get close enough. These sharks are all 8 to 9 feet long and 200 to 300 pounds each. And this is my friend Wes just standing in the water next to them. So shark mating behavior involves the males. The shark mating is very much like mammalian mating. Parents, you'll know what I'm talking about. Except sharks don't have hands, so they have to actually grasp each other with their teeth. They have to hold on. The male sharks will bite the female's pectoral fins, and then mating proceeds with these unique postures and behaviors that are very easy to study with an accelerometer because the data looks so much different than anything that you would normally see in their day-to-day lives. Here's a male shark grasping a female's pectoral fin. We say grasping, not biting, because they can actually control how hard they bite down. You saw what that tiger shark did to that other tiger shark. Imagine a species where every time they tried to mate, the male bit the female in half. That species would not last very long. So they can actually, even with their powerful jaws, they can actually modulate and control, and believe it or not, sharks can be gentle. So here's a video, I promised this to some of the college students, that I would show a video of sharks mating, and here it is. We have a male grasping the female's pectoral fin, and focus on all of this rolling behavior, because that looks fantastic on an accelerometer data trace. Now we have another shot here. The male's grasping the female. In a second, they'll come around, and you can see the male shark has 2 paired claspers, which are his organ that he uses to introduce into the female's cloaca. And you can see this is a successful copulation. And in a second here, they will come apart. This is the only place in the world where you can see shark mating. Everything we know about shark mating behavior comes from this population in the Tri-Tortugas and the Florida Keys. So I wanted to go there and see if we could use our accelerometers to study the mating behavior so we can learn more than just what we see during the day in the shallow water. What's happening in deeper water, what's happening at night. But in order to do that, these tags are great because they produce 100 data points every second. But it's too much data to transmit back to us. We have to actually, the data are stored to memory, we have to get them back from the shark. And we do that by inserting the tag in this float package that we designed and build ourselves. And then we can attach it to the shark's fin with a galvanic link, with a strap that corrodes in seawater and eventually releases the tag and allows it to come off and float to the surface. I'll show you a little bit more about that in a second. But the nice thing is, you can also see we're holding these sharks in a net in the shallows. So nurse sharks are also great because they're very easy to work with. You can restrain them in the shallows for long periods, they're totally fine. And we catch them by sneaking up on them during a mating event. Turns out they're very distracted when they're mating. So you can walk right up to them and dip a giant net over their head and catch them in this giant butterfly net. This is my friend Wes catching one, and then once you have them in the net, you can just hold them and do all your tagging. Now, of course, this can also be dangerous, not because of the sharks, but because if you run too close to Wes with your own net and he swings the handle of his net around, you can get hit in the face, and I actually lost a tooth doing this. So I have a titanium implant. Don't ask about the other missing teeth in dental history going on. Those were not shark related. But the cool thing about this place is that you can tag these sharks and then you can actually observe them swimming around with your tags. So you can use your direct observations to correlate with the data that you're seeing in the tags. So for instance, this is what it looks like when a shark is swimming. You get these spikes in acceleration, 1 spike for every tail beat. And then when they're resting, you get this flat line. Sometimes you'll have a shark that's swimming normally, and then you'll get a disturbance, and all of a sudden it swims away at a faster rate with stronger acceleration. I know for a fact this was a disturbance that happened here because I was the one that disturbed it. I was snorkeling around trying to track it with my underwater receiver. The water was a little cloudy, and the shark and I almost ran into each other. So this is the value of this site. And then, of course, the mating behavior produces all kinds of really unique data traces. Let's see, where do we go here? There we go. So here's data just from raw acceleration data of a mating event. And you can see it just looks really noisy. We record on 3 axes here. But we can filter this noisy data out so that we see just the animal's body posture. The same thing your phone is doing when it flips the picture. We know whether the shark is upright or head down. And then we can filter out and look at just the acceleration that comes from the animal's movement, just like your Fitbit is doing. And we can use this information to tell when and where they're mating. So it turns out that some sharks will mate just a few times a day, like this one. Other sharks will mate many times a day, and it depends on which shark and also what time during the mating season you tag them. Another interesting story is that we nickname all of our sharks when we catch them and tag them. And this shark was nicknamed after my sister. And this was before we saw any of the data. So I have no idea that the shark nicknamed Anna Luther was going to be our world record for most mating events. Thank you for coming. But we were able to use this data and find out what time of day. It turns out most mating events are taking place during the day, at least on the animals that we've tagged so far. Very few happening at night. And most of the mating events are pretty short, 2 minutes or less. Most of the mating events are not successful. Over 92% of the time, the females will actually avoid mating with the males. And they're very specific on which males they choose to mate with. And so the longer duration mating events are probably the more successful ones, the successful copulations. One mating event over 20 minutes, which is also the point where I look at the audience to see who thinks that's a really long time. The next thing I wanted to study was post-release mortality in sharks. So many of you probably know that shark populations are in trouble worldwide. As humans, we kill about 100 million sharks every year, 70 to 100 million. And of course, only a handful of people are bitten by sharks every year, and just normally a couple of fatalities. But the good news is our sharks in US waters we have the best managed shark populations in the world. We do a really good job for the most part. And also many people in the United States get it now. Far fewer are actually killing sharks when they fish for them. They catch them and then they turn them loose. And the tendency is to think that, okay, if we catch a shark and turn it loose, that it swims off and lives a long, happy life. But actually, we know that's not always the case. I'm just going to show you a little video here about how we actually work with these fishermen.
Sharks that are caught and released by fishermen, how many of them actually survive? To do that, we're using these acceleration data loggers. They're accelerometer tags that record every tail beat the shark makes.
These data loggers combine the depth, the pitch, and tail beats so that Nick and his team can tell when the sharks are swimming down, swimming up, if they live, or if they die. A technique that's never been used to study the sharks off this coast. So here's the process. They first catch the sharks, and then on them put an accelerometer and float package.
So when we roll them over like this, they go into a sleep-like state called tonic immobility. Ideally, they just totally zonk out and hold still throughout the tagging process when they're like this. It doesn't always work. This thing starts corroding in seawater as soon as it gets wet, and eventually it allows the whole strap to come off, and the tag releases from the shark's fin and floats up to the surface. And once it hits the surface, this radio transmitter starts sending out a signal that we can hear from up to about 10 miles away. It's even more fun when we put these tags out and then we have to go out and look for them and get them back.
After 24 to 48 hours, the team now has to recover a whole bunch of floating tags.
All right, so that gives you an idea of how that works. We actually recover over 90% of the tags that we put on sharks, which is shocking to me. When we first started doing this and first invented this float package, I thought we'd be lucky to get half of them back. But when we do get the tags back and look at the data, like from this blacktip shark, for instance, the first thing we look at is the depth data, and we see these spikes with an animal going from the surface down to the bottom over and over again. This is pretty common for sharks. And when we look at the tail beats, the acceleration data, you can see we've got this characteristic, these spikes every time the animal's beating its tail. So here's a blacktip shark that swam off and did just fine and survived. But in other cases, we see a different pattern. We see depth that starts out going up and down, and then it sort of bottoms out here. And we see, when we look at the tail beats, we'll see spikes, but then eventually everything goes to a flat line and it stops moving altogether. This is a dead shark. It was caught, released, and died a few hours afterward. So the good news is, for this particular fishery, is that this is pretty rare. It turns out only about 10% of the sharks caught and released by rod and reel fishermen in Florida, blacktip sharks that is, only about 10% of them die, 90% of them survive. So that's probably sustainable in that fishery. But we're now also working with other fishermen, including commercial longline fishermen. And these are guys who go out and put out miles of lines with hundreds of hooks on them to catch as many sharks as possible. And even though usually they're catching the sharks in order to keep them, to kill them, there are some species that they're not allowed to keep, so they have to release them. And when that happens, we will put our tags on them first and see how they do after they're released. And we're doing this with tiger sharks, sandbars, bull sharks, a couple species of hammerheads, blacktips. And you certainly don't need to digest the data on this table, but I just wanted to draw your attention to the mortality rate. So for some species like sandbars, tigers, bulls, you have a very low mortality rate after they're released, 3% up to 8%. The vast majority of those animals are surviving, even after they're caught on longline. But then you look at this other group, blacktips, spinner sharks, gray hammerheads, and you have post-release mortality rates ranging from 40 to 70%. So for those species, even if they are released, there's a very good chance they're going to die anyway. So it just shows that it's very important to go out and do this research. You can't just assume that a species is going to have a certain mortality rate just because it's in a certain fishery or because it's closely related to another species. It doesn't work that way. So finally, I wanted to talk about a little opportunity we've had to work with this species, the white shark. And this came up a few years ago when I started working with a group called O-Search, who uses this ship that has the giant hydraulic lift over the side that they use to actually lift sharks up out of the water. They'll lower it into the ocean, they'll catch a shark from a fishing boat here and lead it up alongside the ship. And then once the shark is over the lift, they'll raise the lift out of the water, bringing up the shark high and dry, and then the scientists get on board and do our tagging. I'm going to show you a little video of how this works. This was a shark we caught off of Jacksonville that became nicknamed Lydia, and is now one of the many sharks that these guys tag that is famous on social media with like 50,000 Twitter followers or something. But this was a cold, cold day in Jacksonville. It was about 40 degrees, and this is her taking the bait and getting hooked. And then the fishing boat will then lead her back to the ship. She's swimming constantly, so they're not towing her, they're just pointing her in the right direction. And they lead her up alongside the lift and get her right over the lift. And then once she comes in, bumps the wall there. Once she comes in, they raise the lift out of the water. Captain Brett here controls the tail, and then once she's high and dry, we scientists get on board. I'm this tall guy in the blue shirt here. We attach our tags, we're taking samples, we're doing measurements. We have water flowing into the mouth and out over the gills so she can breathe while she's out of the water. This is them attaching a satellite tag on the top of the fin. That's my accelerometer down there, the orange tag. And there you can see the water flowing some out of her mouth. We put a wet towel over her head to cover her eyes, which calms her down for some reason. We don't know why. And then when that's all done, she's out of the water for 10 to 15 minutes. The lift goes back down into the water, and she swims off and does her thing. Here are some pictures of some of the other sharks we've tagged with them. This one is probably the most famous one, named Mary Lee. The shark was 16 feet and almost 4,000 pounds, so bigger than most of the sharks in the parking lot. And you can get an idea. These are two actually pretty good-sized adult men up here putting a hose in her mouth, so you can see how big she is compared to them. This is another one. Not sure which one this is, but I'm attaching an accelerometer there. And this is Lydia, who you just saw the video from. So again, we have to get this accelerometer tag back. And when we do, we see an interesting behavior. This is the depth, so the shark's starting at the surface, going down to the bottom, up to the surface again. She does this over and over. But what we see is, when we look at her tail beats, when she's going down to the bottom, you almost get a flat line here. She basically stops swimming on the way down and just glides all the way down to the bottom. When she turns to go up again, you see the tail beats start up. She's beating her tail to propel herself up. So sharks are negatively buoyant. A dead shark will sink, so they have to swim to go up, but on the way down, they can stop swimming and glide. We think this is a strategy for energy conservation. If you imagine being an animal that has to swim every second of your life, from the moment you're born to the moment you die, it makes sense to try and find a way to rest. She may even be sleeping while she's gliding down here. And the last tag I wanted to point out to you that helps us understand the secret lives of sharks is this blue one here, which is a satellite tag. That's on the top of the fin because every time she comes to the surface and that tag comes out of the water when the right kind of satellite is overhead, we get a location from her no matter where she goes in the world. So you get these amazing tracks here. This is Lydia, where she started off of Jacksonville and ended up crossing the Mid-Atlantic Ridge. There was one point where she was closer to France than she was to Florida, where we tagged her. This is Mary Lee's track. You may recall that one of the books I showed you in the beginning that I read when I was a kid, says very clearly that white sharks don't go to Bermuda. Mary Lee had not read that book. Almost immediately after we tagged her, she went straight to Bermuda. She didn't stick around long, but that was fascinating. Sometimes they hug the coastline. Does anybody like to go on vacation in the Carolinas? So do white sharks. But you didn't get bitten, did you? See, it's very, this probably isn't anything new. They've probably been doing this longer than humans have been around, and we managed to be okay. But for a kid who grew up going to the Carolinas and was terrified to go in the ocean, it was pretty cool to be able to put a tag on the shark that inspired the book and the movie Jaws. I just wanted to wrap up now by talking a little bit about what it's like to be a scientist. So, for instance, that study I showed you, that population genetic study that I did on white-tip reef sharks, was published in a scientific journal. They even put a picture of our species on the cover because they liked our paper so much. This is like big time for a scientist. Even though the number of people who have read this paper is probably fewer than the number of people in this room, but that's okay. But I think a lot of people think that scientists do work and then we send it to the scientific journal and they publish it and that's how it works. But in fact, it's a little bit differently. Once I'm done with this study, and it took me the better part of 7 years to get my samples from all over the world and do this project, I analyze the data, write up the paper, and I send it to the editor of this journal. And the editor doesn't publish it. What the editor does is he sends it to 3 reviewers, anonymous reviewers. I don't get to see who these people are. The only thing I know about them is they're not my friends, because they're not allowed to send the paper to my friends. But who they are is they are people who are experts in my field, and they're usually competing with me for grants or for jobs. So they have every incentive to poke holes in my paper and find all the problems with it that they can. And they do. They find lots of problems. And for any of you who know scientists, we are an argumentative group of people, as my sister and wife can attest to. But they write up all these reviews and find every single problem they can find in your paper. And then they send it back to you, and you have to read their reviews, and you have to respond to every single comment they make. You either have to make the changes that would satisfy them, or make a solid argument as to why they're wrong and you're right. After that, you respond to all those comments, it goes back to the editor and back to the reviewers, and they get to take another whack at everything you just said and argue with you again. They also get to read the reviews that their other anonymous reviewers write and find new problems that they didn't see the first time. And so they can then comment again, and this can keep going back and forth until the editor finally makes a decision and decides whether or not they're going to publish your paper. All of this can take 6 months to a year. And then at the end of that, you might end up left with nothing. So my point to telling you this story is not to complain or to make it seem like we scientists work harder or are smarter or better than the average person. In fact, quite the contrary. We as scientists recognize that as humans, our ability to reason is flawed. And that's why we use this system to find our flaws and poke holes in our arguments and get closer to the truth. We recognize what a famous physicist, Dr. Richard Feynman, said, is that the first principle is that you must not fool yourself, and you are the easiest person to fool. As scientists, we recognize this, and so we're constantly not just believing what we think, but subjecting ourselves to this rigorous process where we invite people who are our competitors to poke holes in our argument. So we're recognizing the fact that our hardware, our brain hardware that we're dealing with, we're still using the same hardware that evolved in conditions where there was a predator lurking behind every bush, where fear and paranoia served us very well for most of the time that we existed. Our ability to see patterns, even in places where patterns don't exist, used to serve us very well. How many of you here know your zodiac sign? Every single one of you knows your zodiac sign, right? Ever wonder why we don't use that for like job placement or to match you up with your college roommate? It's because it's pure fiction. It's fantasy, it's not real. But there are a lot of things like that in life now, and the things like ability to see patterns and fear and paranoia served us very well for most of our evolution, but we're now living in a world where they aren't serving us as well. And we have things that are very basic, uncontroversial scientific consensus that we're now losing track of. So it didn't used to be controversial to say things like, vaccines are safe and save lives, or human-caused global warming is real. Mermaids are not. Megalodon is extinct. All of these things used to be, all of those things are things that I've had to debate otherwise intelligent adults about, including the mermaids thing. My point is just, and for those of you college students who may be hearing my message and thinking, you know what, he's right, and there should be a law that, you know, bans people from saying things like that. No, 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 please. That is not the answer. It has to be always allowed for people to speak their ideas, no matter how ridiculous they are. But when they do, those of us who know better have to then speak up and speak the truth every chance we get. Even if you're studying, you know, river mussels or sharks or insects or whatever the case may be, even if it's just in the last few minutes of a talk on the secret lives of sharks. Thank you very much.
Okay, we're going to have some time for Q&A, and if you don't mind, Jonathan will bring